environment. Develop ability for the application of advanced to improve agriculture and nutrition in the country's region. तर हे आपल्याला इम्प्रुव्हमेंट जर करायचे असेल ऍग्रीकल्चर संदर्भात जर आपला असेल दिलेल्या ऑब्जेक्टिव्हनुसार आपण गेलो so these course are complete within 1 year and the pattern of this course is that the semester pattern given by CBCS authority. So with the help of this one, so this is the one paper. First semester. So this first semester have paper one, biodiversity of microbes, algae, and fungi. Paper second, plant ecology. What mean practical? Practical based on theory paper one and second. Semester second, paper third, biodiversity of archaeogoniate, biophytes, pteridophytes, gymnosperm, paper. Paper four, plant taxonomy. And this is a practical depend upon practical third and practical. One minute.
Hello, share screen share दिसतोय का?
Yes.
Hello.
So this is the entire paper the for semester one.
Screen आवाज येतोय का? Hello.
And another paper second plant ecology.
Hello.
Practical based on paper one and paper two.
Hello.
On biodiversity of microalgae and plant ecology.
येस सर
आवाज येतोय का?
आवाज येतोय पण स्क्रीन दिसत नाहीये म्हणजे पीपीटी दिसत नाहीये फक्त स्क्रीन दिसते
स्क्रीनच दिसते परत एकदा
and the practical having based on practical and paper. So this is this
पीपीटी दिसत नाही काय झालंय बघ पीपीटी दिसत नाही
पीपीटी प्रेझेंट
केली
आता दिसत
ए दिसते का बाळा आता
at the time of
हॅलो
स्टुडंट्स इमिजिएटली अनम्यूट युअरसेल्फ
नो नो नाही दिसते
आता दिसतेय का
after completion of the practical and theory.
ए बाळा पीपीटी दिसते का आता
नाही दिसत
नाही दिसत
So in library you can observe the number of textbooks, journal, periodicals available in institute and departmental library. A separate references are attached along with the listed
विंडो कर बरं
so library का सगळ्यांना हॅलो हॅलो
स्क्रीन
हॅलो
परत एकदा आपण करूया
विंडो विंडो
एंटायर हे
अगं विंडो कर की मला इकडे तिकडे घेता येते दोन्ही विंडो कर म्हणजे
आता दिसतंय का
दिसतंय मी हा मॅडम दिसते
विंडो विंडो वरती नेता
हे बघा एस्केप दाखवलं ना हे जे दिसेल डायरेक्ट क्रोम वर जायचं आता
एस्केप पेक्षा नुसतं विंडोवर इकडे तिकडे केलं तर काय
असं पण दिसेल हे बघा इथे गेलं की फक्त इथे जायचं
पण ते एस्केप करायला लागते ना आतापर्यंत
आता असं जरी ठेवलं तरी दिसणार
मुलांना दिसू दे पण मुलांचं परत मला याच्यावर न्यायचं म्हटलं तर परत एस्केप केल्याशिवाय असं का नुसतं असं
तसं होऊ शकतं
आणि परत
हे आता सुरू आहे
in a library we can observe reference to periodicals available.
Okay, am I audible?
Separate reference is
Hello. आवाज येतोय का?
Oh, sir.
Very good, very good. Okay, last time we were discussing about general principles and methods of food preservation. And in that general principles and methods of food preservation, we were discussing about the techniques which are used for food preservation and the principle involved in those techniques. For example, asepsis is one of the technique Asepsis is one of the techniques used for food preservation. Then, in case of asepsis, which take a principle involved that we have seen, that is the techniques asepsis, removal, and anaerobic condition. Then last time we have seen the basically the food which is obtained for human being basically from two sources, that is from plants and animals, and again it is divided into four categories, that is cereal and cereal products, sugar products, vegetables and vegetable products, fruits and fruit products, meat and meat products, poultry, eggs, fish and other seafoods, milk and milk products. Then we have seen classification of food, that is perishable, non-perishable, and semi-perishable food. Then we have seen causes of spoilage. Then we have seen methods of food preservation. Just now I have told you people, that is asepsis, removal, use of high temperature, use of low temperature, drying, etc. All these techniques. And I think last time we have seen the basic principles involved in the food preservation, and that is asepsis मध्ये keeping out microorganism, removal मध्ये filtration principle किंवा removal मध्ये पाहिले, hindering मध्ये drying, anaerobic condition, low temperatures, chemicals which stops or which inhibits, then by killing of microorganisms, heat radiation principle. Then prevention or delay of cell decomposition, destruction, that is inactivation of enzymes involved, blanching, etc. We have seen. And asepsis, म्हणजे लास्ट टाइम मी असेप्सिस बद्दल सांगितलेलं होतं की नेचर मध्ये देर आर व्हेरियस न्यूमरस एक्झाम्पल्स ऑफ asepsis, keeping out microorganisms or preservative factors, that is inner tissues of healthy plants. Animals are usually free from microorganisms, and if any are present, they are likely to initiate spoilage. If there is a protective covering about the food, microbial decomposition is delayed or prevented. And examples of such coverings are natural shells of nuts, skins of fruits and vegetables, husk of ear corn, eggs, etc. Last time we have seen. And then question arises, the naked food is there, the ready-made the cooked food is there, uncooked food is there, food which is ready to eat is there. So that food we can preserve, that food we can preserve by that food we can preserve by carrying out cooking. So packaging of food is widely used application of asepsis. लास्ट टाइम आपण काय पाहत होतो की packaging of the food material is important, and with the help of packaging we can keep microorganisms away from the food material. The entry of microorganism is prevented by carrying out packaging. As far as this packaging point is considered, there is a packaging industry is there, packaging engineering is there, packaging a very large industry is there. So you might have been seen various kinds of food. Say for example, milk powder is there. Say for example, your instant proteinous food material is there. Bornvita is there. Coffee sachets are there. Then various other chocolates are there. So packaging is of different kinds. The main aim, important object of packaging, that is you should avoid entry of microorganisms. Entry of microorganism is prevented by use of packaging. So packaging is most important. So packaging of food is widely used application of asepsis. The covering may be ranged from loose cartoon or wrapping packaging, which prevents primarily contamination during handling to the hermetically. loose cartoon packing to the hermetically sealed container of the can. Hermetically, that is entirely in aseptic conditions, entirely in a sterilized conditions, in a sterilized fashion. Packaging is carried out. This is called as a hermetically sealed. So the packaging starts from loose cartoon or wrapping to hermetically sealed container of canned food, various kinds of food hermetically. So as far as packaging is considered, you remember well, a loose cartoon package to a hermetically sealed package is there. Loose cartoon package to hermetically sealed packaging is there. So basically, this in the canning industry, the load of microorganism determines the heat process necessary for preservation of the food. In meat packing industry, sanitary methods of slaughterhouse, handling methods, and processing reduce the load, thus improve the keeping quality of the meat and meat products. So this is an allied information which is related to asepsis. Basically, meat industry slaughterhouses packing is carried out. So while carrying out the packing, you have to maintain good sanitary conditions, hygienic conditions in the slaughterhouse, which reduces this load of microorganism. Load of microorganism. So this is about the asepsis. This is about asepsis.
137098
झालं का काम? हा? चालेल. All the best. So asepsis झालं. आता removal of microorganism. आता what happens by any means, if packaging is carried out in a proper fashion from loose cartoon package to hermetically sealed packages. So as you are aware, microorganisms are ubiquitous, they are present everywhere, and entry of microorganisms may occur by any way or any fashion. So what happens once entry of microorganism occurs? So before the growth of microorganism, if you remove the microorganism from such foods, you can preserve food for certain period of time. So removal of microorganism, this is another technique. This is another method of food preservation, removal of microorganisms. So this technique, for most part, the removal of microorganism is not very effective in food preservation, but under certain special conditions, it may be helpful, and removal may be accomplished by filtration technique is there, centrifugation is there, sedimentation is there, clarification is there, washing is there, trimming is there. So these are the techniques used for removal of microorganism once entry of organism is there in a food material. Entry of organism is there in a food material. So basically what happens, this removal of microorganism, we can use it for liquid food material. Liquid food material. It is rather difficult to use the principles involved in those techniques, that is filtration, centrifugation, sedimentation, clarification, washing, trimming, washing and trimming. To some extent we can use it for solid food material for removal of microorganisms. But filtration, centrifugation, sedimentation, then clarification, for all these techniques, the food must be present in a liquid form, in a broth form. Then we can use this filtration, centrifugation, sedimentation, and clarification. Washing and trimming, we can use it for a solid food material. Solid food material. Of these, filtration is the only successful method for complete removal of organism. and its use is limited to clear liquids. Just now I have told you people. The liquid is filtered through previously sterilized bacteria-proof filter, which is made up of asbestos, sintered glass, diatomaceous earth, unglazed porcelain. So uh, these are the materials, that is asbestos, sintered glass, diatomaceous earth, unglazed porcelain, various kinds of membranes, that is acetate membrane, then various membranes are there. The liquid is forced by positive or negative pressure, and this method is successfully uh, used for with fruit juices, beer, soft drinks, wine, and water. Wine and water. So all of you remember well, for removal of microorganisms or preservation of the food by removal of microorganism, the technique, one of the most reliable technique, promising technique, that technique is nothing but the filtration technique. Filtration technique, and for this filtration technique, the pore size you have to prepare from asbestos, sintered glass, diatomaceous earth, unglazed porcelain, or from various membranes, and it is used for liquid food, basically various kinds of fruit juices, beer, soft drinks, wine, and water. Apart from this filtration, centrifugation or sedimentation, which generally not effective in that some of the microorganisms removed, but not all microorganisms. Okay, how many of you are there? 54. तर समजतंय का काय बाळांनो? सुचेता, रेश्मा, येस. अरे बाळांनो, मी काय बोलतोय ते समजतंय का?
हो सर.
हा,
हो.
समजतंय ना? Then
मग एखादं एक्झाम्पल सांगताय का? सांगता येईल का?
so washing of fruits and vegetables removes surface microflora from soil and environment. Washing of cabbage heads or cucumbers before their fermentation into sauerkraut and pickles removes soil microorganisms. Trimming, that is cutting away the spoiled portions of food or discarding spoiled samples, is important for removal of large number of spoilage microorganisms. Large number of spoilage microorganisms. आता आपणाला कोणती, आता आपण असेप्सिस आणि रिमूव्हल पाहिले टेक्निक, मेथड्स ऑफ फूड प्रिझर्वेशन मध्ये वन ऑफ द नेक्स्ट मेथड, दॅट इज प्रिझर्वेशन बाय यूज ऑफ हाय टेम्परेचर. प्रिझर्वेशन, आता आपण कुठल्या मेथड पाहिल्या? आता आपण कुठली मेथड पाहिली? जस्ट नाउ वी हॅव सीन विच टेक्निक, that is asepsis or removal of microorganisms. Asepsis and removal of microorganisms या दोन मेथड पाहिल्या. आता त्याच्यानंतर कुठली मेथड आहे? त्याच्यानंतर कुठली मेथड आहे?
हॅलो, याच्यामध्ये काय भाग आहे?
Preservation by use of high temperature. Preservation by use of high temperature. आता मला कोण सांगेल प्रिझर्वेशन बाय यूज ऑफ हाय टेम्परेचर? दिस इज वन ऑफ द मोस्ट reliable technique. This is one of the most promising technique which is used for preservation of the food. Preservation of the food. So as far as this temperature is considered basically last year you have studied control of microorganisms by use of either moist heat or use of a dry heat, that is use of hot air oven or by use of ऑटोक्लेव ऑटोक्लेव तर मला कोण सांगू शकेल तुमच्यातलं प्रिन्सिपल इन्व्हॉल्ड in case of control of microorganisms or preservation of food by use of a temperature, by use of temperature. Who would like to tell me the principle involved in case of temperature? How microorganisms get killed by application of temperature? रेशमा सांगू शकतेस का? रेशमा मला सांगू शकते का? टेम्परेचरचं बेसिक प्रिन्सिपल काय असतं? कोण सांगू शकेल बेसिक टेम्परेचर? Pasteurization. What is basic difference between pasteurization and sterilization? Pasteurization आणि sterilization मध्ये काय फरक आहे? कोण सांगू शकेल? Pasteurization आणि sterilization मध्ये काय फरक आहे बाळांनो? त्याच्यामध्ये इंट्रोडक्शन आहे, जनरल कॅरेक्टर आहे, फॉर्म्स अँड स्ट्रक्चर. कोण सांगू शकेल? पाश्चरायझेशन, स्टरलायझेशन. रेशमा. In both the techniques, temperature is used. Use of temperature. Use of temperature. The temperature may be low temperature or temperature may be high temperature. May be low temperature or temperature may be high temperature. So preservation by use of high temperature. Preservation by use of high temperature. So all of you are aware. All of you are aware. Basically what happens when we apply, there is a always time and temperature relationship is there. Time and temperature relationship is there, and so as to decide, so as to find out, so as to determine, so as to optimize this time and temperature relationship. Last year we have seen TDT, thermal death time, TDP, thermal death point. बरोबर आहे का? लास्ट इयर आपण पाहिलेलं आहे थर्मल डेथ टाइम, thermal death point. That is, in certain cases, the standard time required to kill standard suspension or temperature required to kill standard at a constant time temperature relationship. Time temperature relationship last year आपण पाहिलेलं आहे TDT, TDP, then D value, F value, Z value. So all these values are most important when we apply a high temperature. So basically what happens when you go through this your हॅलो
सर नेटचा प्रॉब्लेम आहे, तुम्ही बोलत बसू नका.
हॅलो एम आय ऑडिबल?
सर नेटचा प्रॉब्लेम आहे, ऐकू जाणार नाही त्याला.
बघू आता. हॅलो
चालू झाला नेट.
हॅलो सर.
येस सर.
येस सर.
एम आय ऑडिबल? व्हेरी गुड. स्क्रीन दिसते का बाळानो? स्क्रीन दिसत नाही. आता दिसते का रे?
हो दिसते.
दिसते ना? व्हेरी गुड, व्हेरी गुड. Okay. So we were talking about preservation by use of high temperature. So in this preservation by use of high temperature, I was talking you people that application of temperature, whether it is a dry heat is there or moist heat is there. So what happens, as all of you are aware, when you go through this chemical composition of the bacterial cells, you will find that there. Yes.
सर तुम्ही पीपीटी
हो.
शेअर केली आहे का? हा मग
अरे
दिसत
आता
नाहीये.
कोणतरी दिसते म्हणाले.
सर स्क्रीन दिसते, पीपीटी दिसत नाही. विंडो सिलेक्ट करा.
विंडो काय करू आता?
विंडो सिलेक्ट करा.
अरे ते स्टॉप प्रेझेंटिंगला आलं.
हो स्टॉप करून पुन्हा स्टार्ट करा. स्टार्ट केल्यानंतर एंटायर स्क्रीन करू नका, विंडोज करा. विंडोज टॅब.
आता बंद केला. आता प्रेझेंट नाव करू का? विंडो करू.
प्रेझेंट नाव करा, त्यानंतर विंडो करा.
आणि विंडो केल्यानंतर शेअर करायला पाहिजेच की विंडो.
हो ते शेअर होते.
आता दिसते का?
हो दिसते आता.
ओके, व्हेरी गुड, व्हेरी गुड, व्हेरी गुड. बर, एक 5 मिनिटात संपूया रे बाळांनो, 5 मिनिटात संपूया. मी सांगत होतो, आय वॉज टेलिंग यू पीपल दॅट इज यूज ऑफ लो टेम्परेचर, यूज ऑफ हाय टेम्परेचर फॉर प्रिझर्वेशन ऑफ फूड. So in the killing of microorganisms by heat is due to denaturation of proteins and inactivation of enzymes required for metabolism. So all of you are aware the microbial cells, when you will carry out the analysis of those microbial cells, you will find that they are mainly composed of carbon, nitrogen, carbon and nitrogen. And this nitrogen is nothing but they are mainly composed of proteins. And as all of you are aware, when we apply temperature to the proteins, there is a denaturation of the proteins. Can you tell, anybody from the class would like to tell me the denaturation of the protein? That is the natural, what happens, the natural structure of protein gets changed. Can you tell me any example of the protein in your day-to-day life that natural protein gets denatured, it gets changed its structure? Anybody from the class would like to tell me the example in your day-to-day life when we apply heat to the protein material in our day-to-day life, the protein material changes its natural structure. कोणी सांगू शकेल का मला तुमच्या डे टू डे लाईफ मधलं एक्झाम्पल की आपण ज्यावेळी प्रोटीनला हीट अप्लाय करत असतो त्यावेळी त्याचं स्ट्रक्चर बदलत असतं. येस, एनीबडी फ्रॉम द क्लास? अक्षय?
सर बॉइल्ड एग.
Boiled egg, very good, very good. When we prepare omelette or boiled egg, when we apply the temperature, the what you can say this natural structure, that is a liquid structure of the protein gets changed and it becomes a solid. And as a result, what happens? So this entire metabolic activities from the egg gets stopped. So what happens here? The same thing I would like to tell you people, the killing of microorganisms by heat is due to denaturation of the proteins and inactivation of enzymes required for the vital metabolic activities. Vital metabolic activities. The heat treatment necessary to kill organisms or spores varies with the kind of organism. This is one of the most important statement. The heat treatment necessary to kill organisms or spores, which varies with the kind of organism, its state and its environment during heating, its state and its environment during heating. The heat treatment selected will depend upon the kinds of organism to be killed. To be killed. And one more point, that is the time and temperature relationship. The time for killing cells or the spores under given set of condition decreases as temperature increases. The initial concentration of cells or spores, previous history of cells or spores, the culture medium, all these points you have to take into the consideration when you carry out the food preservation by use of temperature. That is what kind of organisms are involved. Saprophytic microorganisms are involved. Pathogenic microorganisms are involved. Spore forming microorganisms are involved. So all these points you have to take into the consideration for proper sterilization or for proper preservation. The time and temperature relationship, just now I have told you people, the time for killing cells or spores under given set of condition decreases as temperature increases. That is, then you have to take into the consideration initial concentration of cells or spores, initial concentration of cells or spores, then previous history of the cells or spores, then culture medium. All these points you have to take into the consideration when you carry out the uh, food preservation by use of high temperature. So, of the various techniques, use of high temperature, this is one of the most reliable technique. This is one of the most promising technique which is used for food preservation. Promising technique for food preservation, use of high temperature. So again, apart from these, you have to take into the consideration temperature of incubation, temperature of storage. This also you have to take into the phase of growth or age. Phase of growth or age. The as all of you are aware, lag phase is there, log phase is there, stationary phase is there, and decline phase is there. For the application of heat, whether you are applying it in a lag phase, log phase, more effective. Then which kind of heat is used? That is moist heat is used or dry heat is used. Then hydrogen ion concentration, that is pH above 5.3, pH between 5.3 to 4.5, acid food is there, high acid food is there. All these points you have to take into the consideration, and for all these points they have been mentioned, some of the what they have been mentioned, some of the organisms which will remain, etc. And just now we have seen, that is pasteurization, sterilization, then thermoduric, thermophilic. So the pasteurization is the technique that is mostly used for milk. For pasteurization is carried out, and this the time and temperature used in case of pasteurization is at 62.8 degrees centigrade for half an hour, and this is every particle of milk is heated at that temperature and immediately cooling at room temperature. We call it as a pasteurization. And sterilization is the process of complete killing of, complete removal of, that is saprophytic, pathogenic, and spore-forming microorganisms. Whereas in case of pasteurization only, pathogenic microorganisms are removed. Pathogenic microorganisms are removed. And as far as pathogenic microorganisms are considered, amongst this Mycobacterium tuberculosis, which can cause the tuberculosis, and Coxella burnetii, which causes this Q fever. So amongst pathogenic microorganisms, these two microorganisms are the most heat-resistant microorganisms. And these two heat-resistant microorganisms gets killed at the temperature of the pasteurization. That is below the pasteurization temperature, that is below 62.8 degrees centigrade, below 60. So there is a chance or possibility of the question may be asked, index organism of pasteurization, bacterial or what you can say, this rickettsial Coxella burnetii organism. Then you should be aware of nature of the organism, that is a thermoduric, survive at relatively high temperature. Some of the microorganisms can tolerate the high temperature, that is up to 55 to 65 degrees centigrade, and we call it, those organisms are thermoduric microorganisms. And in a similar fashion, thermophilic microorganisms are there. Those survive and grow at high temperature. Then this is aseptic packaging is there. Aseptic packaging we would like to see, next time we will see aseptic packaging. So what happens, all of you remember well, of these various food preservation technique methods, you have to remember well, one of the most satisfactory, one of the most promising, one of the most reliable technique of food preservation is application of the high temperature. So do you have any problem in understanding? Will you able to write? Yes, will you able to write? Yes. प्रीती जाधव लिहिता येईल का बाळानो? येस. ओ माने सर, या की जरा इकडं. येस. आता काय करायचं?
मेंबरचा तास सुरू झाला.
आमच्या
मग काय,
आमच्या काय माहिती नाही. हे काय करायचं?
हे दाबण धरायचं
आणि काय येतं मग? हे CTRL आणि काय?
पिक्टारेल.
कुठं आहे PTRL?
थांबा, तेव्हा इथं दाब टाकवायचा
आणि
कुठे तेव्हा हे दाबायचं. सर आहेत की, सर सांग.
दाबलं की आता कुठं काय आलं?
कुठं स्लाइड
ठीक आहे.
केलंय मी आता. थांबा,
हे झाले, ठीक आहे. इथपर्यंत आलंय.
इथपर्यंत आलंय.
ही स्लाइड टाकायची.
इथं कंट्रोल V करा. कंट्रोल आणि V.
करू का?
त्या सोडून
काय काय व्हायचं? काय सांगतात? हे सगळं आलं वाट. कंट्रोल
ढिगभर आलंय.
काय ढिगवर आलं?
स्लाइड सगळे कशाला पाहिजे? किती वेळा दाबलं तुम्ही?
एकदाच दाबलं, जास्त वेळा घेतलं.
मग एवढंच काय आलं?
काय माहिती आता बघा. अंडू कर. काय कर?
आता परत करायचं. कंट्रोल आणि हे दाबत. हे
सुरू होता.
ह्याच्यावर जाऊ दे. थांबा. तुमचं घेतलं. हात काढा. परत करा.
कंट्रोल आणि PT हेच ना?
हा, तेच. बस द्या सोडून.
आता कंट्रोल V
करा. कंट्रोल इकडं सेव्ह करायचं.
कुठं सेव्ह करायचं?
तुम्ही 2 वेळा दाबताय सर.
चालेल.
मग
आलाय आता.
हे ताची तारीख टाका.
आज
हा प्रूफ आहे ना. काय अटेंडन्स बनायचा आणि तारीख काढायची. आपण ही माहिती सांगितली.
ऑटोमॅटिक काय कोणी मला दररोज वेगवेगळं तुम्ही सांगताय सगळे. ऑटोमॅटिक जातो. आता सेव्ह करायचं का परत माने सर इथनं?
स्कॅन करायचं तिथं जोडा.
नुसतं सेव्ह म्हणा का सेव्ह? सेव्ह म्हणा. हे कुठे की जात असतं सेव्ह? आता बंद करू. काही अगोदर बंद कर. ऑफ शेअरिंग.
A very good afternoon to all of you.